then they will generate the allolactose as an inducer. Then they start the inducer will be attached to the inhibitor, which is called the repressor, and they change the morphology, and they do not attach to the lacto-O, and RNA polymerase attached to the promoter, then they start to do the transcription. Therefore, beta-glucosidase, permease glucosidase, transacetylase start to translate and transcription and translation. They start to work. So that's a very simple scenario. By the way, this is what happened for tryptophan operon. Okay, tryptophan operon is originally they will not attach to the O. So RNA polymerase attached to the promoter, the tryptophan will biosynthesize. But once the tryptophan is biosynthesized to a certain level, they said it's enough. We need to do something. We need to stop to generate tryptophan. So at this moment, the tryptophan is a co-repressor because they attach to the repressor, become a co-repressor. They are back to attach to the O, so the transcription will stop. So you can see here, lacto-operon is for catabolism, which means use lactose, become glucose and galactose. Tryptophan operon is opposite direction, is biosynthesized tryptophan. Once it's created enough, they will say it's enough. Don't do it anymore. Why they have to do like this? Because bacteria is very short lifetime. They have to be very economically to handle the situation. So that's a two different way to talk about tryptophan. So Menard and Jacob at first, when they did research for lacto-operon, it just stopped here. But later on, people find some of the scenario which is interesting. What happened is, they find in the bacterial medium, if there is glucose and there is the lactose, it is very interesting. So remember the growth curve. This is time, and this is the bacterial population. They find they come up with two different growth curves, which is at the beginning it looks like that, then it grows again. This is we call dioxic growth curve. And later on, people find it is very interesting when the glucose and the lactose coexisting. Bacteria use glucose first, then they use lactose. So this comes out with another thing. We talk about here the lactose is only talk about this guy, which is the downstream, that is negative control. We haven't talked about here, haven't talked about the cap. We haven't talked about the positive control. And with this situation happens, this means bacteria use lactose or not also depends on the presence, absence of the glucose, which means they also regulated by positive control. So let's talk about that one. This is very interesting because this is need to talk about the knowledge we have already learned previously. So we're going to talk this. We're going to take this out. Okay. We're going to take this whole thing out, and you already know here. So let's talk about something. We're still going to draw this. We have three sides. We have P, O, lac Z, lac Y, and lac A. And this is A. This is CAP position. we already talked about this section. We haven't talked about here. Okay. This is something we need to say. Lactose operon have to work based on the research, rely on glucose and lactose. This comes back to the original question when we learned in our exam 1. The slides will show you how bacteria transfer glucose inside. Let's talk about that. I have a bacteria double fluid mosaic model right here. You still remember, I talk about this all the time. Right here. Okay? When the glucose comes in, and I said it very clear, when the glucose comes in, for bacteria, they become glucose 6-phosphate. But in bacteria, they are not going through the enzyme called hexokinase. They are going through group translocation, and we talk about that. Now what happens? So you have a pyruvate generated. Pyruvate will have 1 phosphate, okay? The phosphates coming, we're going through is enzyme 1. And then they will going through is HPR, heat stable protein. We talk about that. And finally, where they're going to go is the enzyme 2. The tricky thing is here. If there is a glucose, the glucose is present, they will go this direction. So let's try a little bit more. Try a little bit more. They will go the other direction. Because here, you have N9 complex 2, enzyme complex 2, AB complex. So they go this way. They here become glucose 6-phosphate. They go inside of the cell memory system. If there's no glucose, what happened? They're not going to go this way. Instead of, they activate another magic enzyme here. It's called AC. What is that? Adenyl cyclase. Okay, when this adenyl cyclase happened, the ATP will become CAMP. This is something you should know very clearly what will happen from the chemical structure. ATP becomes CAMP. What will happen? So we talked about this already. Yes. If you have a structure here, okay, then H2, and N, Then you go here, we have phosphates, and H, OH, H, OH. Then go here, we have H, CH2. You go here, O, phosphates, O, phosphates. And you go here, O, phosphates. That's ATP. What happened if you have adenine cyclase? These two are gone. This is gone. This connected with here. This is CAMP. Cyclic adenine monophosphate. When the cyclic adenine monophosphate generate, they will attach to, sorry, not on top, they will attach to cap. So once the CMP attached to the cap, the transcription will happen. If the CMP is not attached to the cap, the transcription will be stopped. So, this comes out with something is, we want the lactose operon to work. The prerequisite requirements is in the environment, there is no glucose. Because if there is a glucose, they will generate glucose 6-phosphate. They will not activate adenine cyclase. So this reaction will not happen, which means this ATP will not generate the CMP, therefore the CMP will not attach to the cap, and the transcription will not happen. And no matter what happened here, and I gave everybody a joke. This is like you get married, you get a pressure from your parents. They will say you need to have a baby in the second year, blah, blah, blah. Then you get a pressure from your mother-in-law and the father-in-law. Because they may say, we don't have enough money, you need to be treating your wife better. That's the same for the bacteria, to using the glucose and the lactose, although they cannot get married, but when they choose a different nutrition ingredients, it's the same story. So with all being said here, this comes out with a very tricky and puzzle situation. So we're going to draw the situation right here, okay? This is come out with a tricky situation. Because it could be attached or not attached to the cap position. They could be attached or not attached to the O position. So the possibility is 2 of each. 2 multiplied by 2 equals 4. This comes out with 4 different scenarios for lactose operon. So what are they? We just still draw it, okay? CAP, P, O, lac Z, lac Y, lac A. Let's draw all these 4. Let's talk about that. Cap, P, O, lac Z, lac Y, and lac A. We draw another one. I hope you can draw it on your notebook. So we'll talk about that. The 4 different situations. Y and lac A. And go here, we're going to talk about the same thing. Is cap, P, O, lac Z, lac Y, and lac A. So let's talk about the first scenario, which means there is glucose and there is lactose. What will happen? So let's talk about it. If there is a glucose, what will happen? Let's talk about there is a lactose. Both are present. Okay. Lactose presented. The inhibitor will not, the tetramer, will not attach to the O. They will be removed. The reason is the lactose will generate allolactose. They are gone. So, right side, the positive control, there is no problem. But how about the glucose? If there is a glucose present, what will happen? The glucose 6-phosphate will generate. They will not generate cap. So, cap, CMP will not connect to the CAP. Is this going to work? No. This transcription will not work. No transcription. This is the first scenario. Okay, the second scenario. If there is a no glucose, but with lactose, The lactose is in. What will happen? Okay, if no glucose, instead of the phosphate transfer from glucose through group translocation, become glucose 6-phosphate, they will activate adenine cyclase. They generate cyclic adenine monophosphate. Therefore, the CAMP will connect to the cation. And secondly, because there is a lactose, so what happened? The inhibitor will connect to the allolactose, and they will be not attached to the operator, which is O position. So the transcription is happening. There is transcription. Yes, it works. That's the second scenario. Third scenario. There is a glucose, but there is no lactose. What's going to happen when this situation happens? If there is a glucose, they will become glucose 6-phosphate. So the lacto-operon will not work it because the CAMP cannot connect it to the CAP. And no lactose, this inhibitor will be the repressor, or inhibitor will attach to the operator, which means double control. They don't let you do anything. So, no transcription. Okay? No transcription. In other words, at this situation, the bacteria can only use glucose and goes through glucose 6-phosphate to become fructose 6-phosphate, fructose 1,6-biphosphate, and glyceroldehyde 3-phosphate going through what we call glycolysis. Okay, that's the third scenario. Last scenario, there is no glucose and there is no lactose. Well, this is like a joke I always say. Of course nothing happened. Because you don't have anything, it's like a distilled water. But what's the mechanism behind it? There is no glucose. Okay, glucose 6-phosphate is not generated. They are instead of activated, become adenine cyclase, become cyclic adenine monophosphates. So, yes, this is good. CAMP is connected to that. However, on the negative control side, because no lactose, so allolactose will not be enough. So what happens? The inhibitor will connect to it. So the repressor, tetramer, will attach firmly to the operator, so there is no transcription will happen. This transcription will not happen, so no transcription. So this sounds like a maze. Okay. Now something else I want to talk about that. How they started to do. The P and O position in the real life, it is not like this. This is just like a model. It will be like O1, P, O2, P, then O3. It's alternative arranged. And it's not linear. It's sometimes a little bit cyclic. So, what happened is when the transcription happens, is actually there is a connection between O1 and the promoter related to the alpha factor of RNA polymerase. So we talk about the RNA polymerase alpha 2 to alpha unit beta, beta prime, omega. This is related to the alpha factor. Okay, so this is important. This 1, 2, 3, 4 situation, you need to remember very well because it is important, important for the understanding these things. Okay, so we can go back to see the slides, what it looks like. We call it a catabolic repression. So this is a slides clearly showed you when the presence of glucose and lactose, there is a secondary growth curve generated, which means bacteria can survive in a very longer time. And we call it a biphasic growth, dioxy growth. So you have the glucose curve, the glucose used is lactose. So this tells us bacteria use glucose first, then they use lactose. So the further research we find that we talk about here is only the negative control. We didn't talk about the positive control because this is only talk about on the right side, which is the downstream. We don't talk about the upstream yet. So this comes with the upstream. And we say the A position is catabolic activated protein. The regulating response to presence or absence of the glucose allows for professional use of the glucose. And this comes with what happened for adenine cyclase. And we draw the picture here, is the same I draw there, is ATP, 5' adenine triphosphates. And once you have adenine cyclase, the two phosphates will gone, then become CAMP, cyclic adenine monophosphates. This is another scenario. Looks like the picture there we talked about already. In our exam 1, lecture 3, we talk about the nutrition translocation, and I said very clearly, the bacteria does not rely on passive distribution, facilitated distribution, and also active transport. They are highly rely on is group translocation. The reason is, if you have a glucose, they directly will become glucose 6-phosphates. But if you don't have a glucose, the phosphate, when they transfer to complex enzyme 2A, they will go to downstream, the down position, activate AC, which is adenine cyclase. Therefore, ATP becomes CAMP. That's what the story comes out, the CAMP connected to the cap. And this is a very good slide, which is connected to knowledge from our exam 1 to so forth. If you don't really understand, this is a simple picture. Low glucose, the adenine cyclase will be activated and become CAMP, then CAMP connected to the cap, so RNA polymerase connected to the promoter. But be careful, this catabolic activator is only talk about is a positive control. In these slides, they did not have a negative control, which is we talk about in the previous couple of slides yet. So this is kind of like confusing you. This is only talk about is cap to CAP. And this is, we just talked about. The regulation of a lacto-operon by the lacto-repressor and the CAMP. Okay? So lactose but no glucose. This situation is the first one. So what happened, the CAMP goes there, repressor is inactivated, transcription happens. Second scenario, there's a lactose, there's a glucose. Repressor is inactivated, but the CAMP cannot go to, CMP cannot go to the CAMP, so no transcription. The third scenario, neither lactose nor glucose is there. Well, CMP go to the CAMP, CAMP is good, but repressor also attached to the operator, no transcription. The last one, glucose but no lactose. CAMP is now working, but no lactose, and they also attach to the they also attach to the operator. Okay, so this looks like a maze, but it works well to explain the lacto-operon, and the knowledge for the positive control, negative control, also relate to the glycolysis, relate to the ATP structure, CMP, we talked earlier. So that's lacto-operon is a very interesting topic. I always like to spending roughly 15 minutes to talk about that, because it is important to connect with all the knowledge here. And also, it using E. coli as an example. That's fit for our mission of the class. We always talk about E. coli. Even E. coli, we're going to do the E. coli study very soon for the generic E. coli. We always talk about that because it's a basic tool to talk about the bacteria. Okay, very quick talk about the restriction enzyme. I'll just use slides. Restriction enzyme is a magic operational tool during the slice and modification of genetic information. They will generate what happened is sticky ends. They cut, recognize sticky ends. So left side GAATTC, right side GAATTC. So when you look at this, do you remember in the lab what we talked? Using IMVC test to differentiate E. coli and Enterobacter is positive, positive, negative, 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 positive, positive. Does that look similar? That's something we talk about that. It's a restriction enzyme. Okay, that's why that biochemistry reaction is very important because very similar to the restriction enzyme to have the sticky ends cut. Now, this is what happened for the cloning. So Jackson, Simmons, and Berg in 1972, they have this recombination of the DNA molecule. So what happened is they using restriction enzyme to cut a certain sequence, which is sticky ends, and then they have a vector, and they can do the recombination. And this is very interesting when they do the recombination. And they are using is E. coli restriction enzyme 1. This one is being studied a lot. So talking about this vector is interesting. They must have some places can be recognized by the E. coli 1, which means they must have a sticky ends. And they also need to have a cloning site, which means foreign DNA can integrate to it. Once they integrate to it, what happens? The function will be gone. So he did a bunch of study. What happened is the LAGZ gene, which is responsible for beta-glycosidase. He used restriction enzyme, which is E. coli 1, to cut it. Okay, when they cut it, what happened? The LAGZ gene is gone, which means beta-glycosidase is gone. So, which means lactose is not break down, become glucose and lactose, galactose. So what happened is that this could be verified in the agar. And when you go to see the agar, you see those blue colony. What it means? They have a vector only, which they means they did not successfully recombine. And when you see those white colony cell, which means the cloned DNA is inserted. Why? Because the LAGZ gene is taken out and inserted the new gene, so the reaction doesn't happen. So that's why the right colony is showed up. And the vector colony, the blue colony, which means the LAGZ is working, and the lactose breakdown become glucose, and in this specific agar, it turns the blue colony. Okay, so this is briefly what I have so far. We don't want to talk about this too much, but in the exam, you should know the conclusion of this slides. Okay, so how to understand this? It's very simple. If no glucose, no lactose, of course nothing happened. If no lactose, of course nothing happened. The lactose operon working, there's only one condition, is no glucose, but lactose present. It sounds like a tricky, like a maze, but actually it's reasonable. Is that right? So that's all I have. Okay. Last, we want to talk about the extra points. Okay, what extra points we're going to offer you? This is very important, so everybody have to listen. What extra points we're going to offer you? It's a tons of. Number one, glycolysis cycle. As part of the aerobic respiration, chemical name plus structure. Number 2, TCA cycle. Part of the aerobic respiration, chemical name and structure. Number 3 is all side chain of 20 amino acids. We will talk about this next week, Tuesday, in the lab section. So the lab one, I will make a video to go over it and put on the campus and you can study it. Okay? That is the one. Chemical and structure. So for example, glycine, alanine. This is 1 point, this is 0.5 points. Make sense? Number 4, ATP structure, 5 points. Number 5, A, T, G, C, U structure. You need to write the full name. Adenine, thymine, guanine, cytosine, uracil. The same thing, chemical name and structure. Okay? This is very important. This why gives you so many things. The idea is you don't give up. We have students haven't done very well in the exam 1, did even worse in the exam 2. Exam 3 is difficult relatively because genetics and biochemistry, relatively speaking, but we still have a practice exam and review. We give you tons of these extra points. This is only you need to work. Is that right? You don't need any wisdom. Your IQ can be only 50. You still can know the answer. Your IQ can be 200. If you don't study, you still don't know the answer. This is test your attitude. Whether you study or not test your intelligence and wisdom. And this is what I like to do is you study the time, spending the time, put your energy there and get the grade what you deserve. So that's give you tons of opportunity. Will give everybody opportunity moving forward for the exam 4. Which means theoretically in this classroom, everybody you still have a chance to get A or A minus if you do the calculation. How many points are there? Maybe 50, 60. Okay? That's good. And how are you going to handle this? It's up to you. It's not required. If you think everything you did very well, you don't have to do anything. Okay? That's all I have.